Alright, sorry about that. I'm having to make this 2 videos because I hit the wrong button on my previous slide, which ended the video. So my apologies, but we're going to pick up still where we left off. We're not starting back over. But anyway, so what we left off with was discussing recombinant DNA technology, and I was showing all the illustration of how we are able to generate bacteria cells that contain human genes for insulin and thus harvest insulin from them. So here we have a human cell. Obviously, this is much more simplistic than a true human cell because if this was illustrated appropriately, it would have had 46 total chromosomes. It's only showing you the chromosomes that have insulin, the insulin gene specifically, right here. So what we will do in the human cell is we're going to remove that insulin gene. We're going to purify our sample so we only have human insulin gene. All that other genetic material will be discarded. Next, we're going to have a bacteria cell. This is our bacteria. Here is the plasmid. It's not showing you the other DNA within our bacteria cell because they are going to have one very large single circular loop of of DNA, and then they'll have some small accessory rings. This image simplified it to just show you a single plasmid. So we're going to remove that plasmid away from our bacterial host cell and cut it with restriction enzymes. That restriction enzyme will allow for that insulin gene that we cut out of the human genetic material to be inserted and ligated into that gaping spot right there. All right, so now that we have our insulin gene inserted into the plasmid, which is seen here, we call that recombinant DNA because we have recombined DNA. We've mixed it together. We have the human insulin gene here and the plasmid DNA in green. So next, we're now going to take that recombinant DNA and reintroduce it into a bacterial host cell. So now as that bacteria cell grows and reproduces, it's going to create other cells that also have the same human insulin gene because it's a DNA polymerase, RNA polymerase. They're not picky. They don't know that they're copying human DNA or bacterial DNA. They're not picky. They're going to copy and paste everything when it comes to replication. They're going to transcribe and translate everything when it comes to protein production. So as this cell multiplies, remember mitosis, 1 becomes 2, 2 becomes 4, 4 becomes 8. All of those new cells are also going to have that human insulin gene. And therefore, all of the cells that descend from the cell are also going to produce human insulin protein. So we harvest that insulin, indicated by the blue dots, into product that is then able to be marketed and sold as a medical supplement. All right, so how is that relevant? Well, you probably are familiar with the term diabetes. So diabetes is a very common, relatively anyway, condition in humans where patients can't make their own insulin and they have to get insulin injections throughout the day sometimes or periodically in order to stay healthy. So what insulin actually does is it helps control your body's sugar, your blood sugar. So when you eat something like a juicy cheeseburger or whatever it is you're eating, it's going to be broken down ultimately into amino acids, glycerol molecules with fatty acids. Or glucose or some kind of simple sugar. Remember, things that you see on a nutritional label: proteins, carbohydrates, and fats. All those things get broken down into their monomers, like we discussed in chapter, I believe it was 3, and then taken to the blood. So at that point, you have glucose, sugar in the blood, that will then be taken to the tissues. So when you eat that juicy cheeseburger, it's going to make your blood sugar spike. Now, as time goes by, your blood sugar should go back down because that sugar should be removed from the blood and taken to the body's tissues, the cells, which need that sugar to do cellular respiration and make ATP. So in people that have diabetes, they are not able to do that. Their body doesn't make enough insulin, and therefore the sugar stays in the blood, and that's why they have high blood sugar. Now, previously, not too terribly long ago, pigs were commonly utilized to harvest insulin. But now we use that biosynthetic human insulin that I described to you just a few seconds ago that derives from bacteria. And so this is another image that shows you the same thing. So just as a reminder, we have our human cell. We take out the human DNA that contains our human insulin gene. We use restriction enzymes to cut away that human insulin gene and purify it, the sample, so we get rid of everything else. Then we have our bacterial cell. We take out a plasmid, which is an accessory ring of DNA found in bacteria, and we cut it with that same restriction enzyme. So now these two sticky ends match the two sticky ends on the insulin human gene. Then we induce the human insulin gene to be ligated into our bacterial plasmid by using DNA ligase. Then we call it recombinant DNA because we have recombined it. It's from two separate species, human and bacterial. Then we induce our bacterial cell to take up that genetically modified recombinant DNA, and then we let the cell do its thing. It starts producing more cells that also have that same recombinant DNA, as you can see. More cells being made, same recombinant DNA is within it, and they all are capable of harvesting or being harvested to get their insulin product that they're making. Okay, the second technology I want to discuss with y'all is polymerase chain reaction, aka PCR. PCR is probably one of the most common biotechnologies utilized in tech labs throughout the world. I've never seen a lab that doesn't have PCR utilization somewhere in their protocols. And the function of PCR is to amplify DNA. And when I say amplify, it means to make copies. So oftentimes whenever we are needing to do something in biotech, having just one cell that has, one full copy of genetic material isn't enough. We sometimes, or very frequently actually, need to make lots of copies, like billions of copies. And that's what happens when we utilize polymerase chain reaction. Polymerase chain reaction, or PCR, utilizes a special DNA polymerase made from, it's harvested from a species called Thermus aquaticus, which is a bacterial cell that lives in high temperature environments, and thus their DNA polymerase tolerates high temperatures. Our cells would not tolerate those high temperatures. Another thing that's utilized in PCR are primers. So we've talked about primers previously. Remember, these are little tiny pieces of DNA, or sometimes RNA, that tells an enzyme where to begin. Remember, we talked about primers before in our previous chapter. So if you need a reminder of what primers are, go back and look at that because it was important. So specific DNA segment to be amplified. The primers help tell DNA polymerase where to go, where to start. And thus, we are not amplifying all DNA. So if I were to put one of my cells into a little test tube and utilize PCR on my genes, I could be specific about what genes I wanted to copy by utilizing primers. Because remember, primers act like little red flags that say, hey, start here. This is where you need to start copying. So go back and look at replication in our last chapter if you need some reminders. So anyway, polymerase chain reaction will cycle over and over again, doubling the amount of DNA after every single cycle. Let's look at the method. Some more of that methodology. We'll look at that in the next slide. I got ahead of myself. Excuse me. Another technology that is DNA sequencing. So DNA sequencing, I mean, it's pretty obvious what happens in DNA sequencing, I think. we're sequencing the DNA. We're determining what are the orders of A's, T's, G's, and C's in a segment of DNA. And that's really cool because what that can do for us is allow us to identify alleles associated with disease. So say I had, hypothetically, sickle cell anemia. A researcher could analyze my DNA and look at my sequence of A's, T's, G's, and C's that's responsible for my hemoglobin protein and see how it differs from normal people. So that's how we associate certain alleles with diseases. It could also aid in forensic biology. So if we have a crime scene sample and I'm a potential suspect, we could analyze the DNA found at the crime scene, compare it to my DNA, and determine if I am a match. DNA sequencing can also help us understand evolutionary history, and that's because the more closely related organisms are, the more closely related their DNA sequences are. So, for example, if you were to compare humans and chimpanzees, their DNA is about 99 point some odd % similar, whereas if you were to compare the DNA of humans versus a banana, then it would be, 40-something% similar. That number was random, but it would be much less than 90% because they're not even in the same kingdom as we are. We're in the animal kingdom, a banana would be in the plant kingdom. Evolutionary history can be determined to an extent by DNA sequencing. All right, so regardless of which one of these applications we're using sequencing for, is very common that, again, you'll see that one of the first steps of any of these processes is going to utilize polymerase chain reaction to make lots of copies. So the way polymerase chain reaction is accomplished is using a thermocycler. A thermocycler, let's break that down, thermo and cycle. Thermo means temperature, and cycle means in a cycle. So a thermocycler alters temperature in a cyclical manner, and you're able to program these thermocyclers to do whatever you want. You could tell it, I want you to change the temperature, and by the way, the samples are loaded here. I want this machine to change the temperatures to 95 degrees Celsius for 30 minutes, and then jump down to 70 degrees Celsius for an hour, and then I want to jump it back up to 78 degrees Celsius for 4 hours. And it would repeat phase 1, 2, and 3 over and over until you told it to stop. So you could tell it that I want you to do it for 30 cycles or 50 cycles, whatever it is that you program it to do. All right, so let's look at the methodology that's going on within it. So once we've extracted DNA from a specimen, again, pretty much any of these biotechnologies that we're discussing, we're going to want to perform PCR to make lots of copies. We're going to need to throw some ingredients into a little test tube to do this. Because again, I'm going to back up for a second. You see all these little things here? These are little wells, little holes that allow for these little tiny tubes to fit in that look kind of like that. I'm not a good artist, but they're little tubes. And the things that we're going to add to those tubes we'll call ingredients. And they're the same things, for the most part, that we needed for DNA replication, which isn't surprising, right? Because, there, that one's a little bit better. Because what we're ultimately trying to do is replicate DNA, just not inside of a cell. We're trying to do it in a test tube. We call that, by the way, in vitro, which literally means in glass in Latin. But anyway, we're trying to do it into a little test tube. So we're going to need the DNA to be copied. So here's my DNA. We're going to need primers. We're going to need free nucleotides, which are free A, T, G's, and C's. And we're going to need our handy-dandy DNA polymerase protein enzyme in order to accomplish all these goals. We're going to add all of those things into this tube. So the first step, once we get everything in the tube, we're going to sit it inside of the thermocycler, and we're going to program it to do the following. We're going to first program it to heat up to about 95 degrees. That's going to cause the first step to occur, which is denaturation. If you recall, denaturation means the breakdown of something. And what it's particularly doing here is it's separating our two strands of DNA. And it's doing so by breaking the hydrogen bond. So if you'll recall, here's just an example. This would be TCAT. So again, this heating up is breaking the hydrogen bonds here, separating our two strands of DNA. So now we have that, okay? Because the hydrogen bonds are, recall the structure of DNA, hydrogen bonds are holding these two strands together. So heating it up will break those bonds, those hydrogen bonds that are holding it together. Now you might be thinking, well, what about DNA polymerase? Remember, that's a protein, and high heats are going to cause them to denature as well. Well, that's not the case with this particular DNA polymerase that we added into our test tube, because remember, it comes from Thermus aquaticus, which is a bacteria that is able to withstand high temperatures. So it does not have any effect on our DNA polymerase. The second step is annealing of primers. So in order to do that, we'll program our PCR to cool down to about 50 to 60 degrees Celsius. So once that occurs, our primers will be added onto our, I'm going to write them as, where's my mouse? As P's. Primer. All right, so they will hybridize to the template strands of the DNA. And I just realized I put DNA. My apologies, DNA. So that lower temperature allows the primers to anneal to the template strands. And then the third step is elongation. And to complete that task, that phase of PCR, we will heat the thermocycler back up to around 72 degrees Celsius. That allows for our DNA polymerase to recognize the primers and then begin adding free nucleotides to the template strands using complementary base pairing rules. And then lastly, T. All right, so again, all these steps occur in response to simply temperature change. So it's really magnificent, really mind-boggling that we can do all of this stuff, and it's so simple, quote unquote simple. You know, the theories behind it are really, really simple anyway. All right, so this image here is going to take you through those same step-by-step procedures, but I wanted you to see their illustration because my drawings are not very good. So here we have our DNA strand that we are, that there's a gene on that we want to copy. So you can see this double-stranded molecule here. So the gene we're wanting to copy is right there. So we're going to use restriction enzymes to cut everything else away. And so now this little gene here is blown up really big. This is our target sequence, what we're wanting to copy and paste many times to make lots of copies. So in the first step, the sample is heated up, about 95 degrees Celsius. That's near boiling point, but not quite. Oops. So once it's heated up, it will separate these two strands. We'll cool it down then in the next step to around 50 to 60 degrees Celsius, which allows for the primers in green to anneal to the DNA templates. Then we'll heat the thermocycler back up to around 72 degrees Celsius. This happens to be the optimal temperature for Taq DNA polymerase. And they call it Taq DNA polymerase because it came from Thermus aquaticus. So we get T and AQ. That's where they get Taq from. Taq DNA polymerase to extend the primer. So Taq DNA polymerase is shown here. And step 3 is the little orangey brown reddish colored ball. And you can see they're going to start extending in both directions, depending on which strand it's elongating. And again, it does that by adding in those free nucleotides that we added into the test tube. We added in some thymines, we added in some guanines, cytosines, and some adenines, thymines, guanines, and cytosines. Okay, so after the first cycle is complete, we started off with 2 strands. Now we have a total of 4, so we doubled it. Then after the second cycle, we'll have 8, and then after the fourth cycle, we'll have 16. And so in a relatively quick amount of time, think an hour or so, we can have millions or sometimes billions of copies of the original target gene that we wanted to copy. All right, so here's a video. I'm hoping it'll be loud enough for you to hear.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. Uses for this technique include creating recombinant DNA, increasing the amount of DNA for forensic testing, or amplifying mitochondrial DNA to determine evolutionary histories. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal-stable DNA polymerase called Taq polymerase, and specific DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA, using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, 1 molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction in a thermocycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
All right. So another technology I want to discuss with y'all is DNA analysis. So another term that sometimes is used synonymously with that is DNA fingerprinting. Previously to super modern day biotech, we used restriction enzymes to cut DNA. And then we would run it on a gel electrophoresis machine, which would analyze the different. It would size separate the DNA fragments that the restrictions enzymes would create. So if you think about it, if you compare my DNA to your DNA, it's not going to be exactly the same, right? We're not genetically identical, unless we were twins. Which we're not, because I don't have any siblings or twins or anything. So anyway, you and I, our DNA is different. So if I were to utilize, let's say EcoR5, which is one of the restriction enzymes on my DNA, and then you were to use the same restriction enzyme on your DNA, and we were to analyze the fragments by size, we would have different size fragments because our DNA is different. And we could analyze that by utilizing a gel electrophoresis box. And the function of the gel electrophoresis machine, you'll get to do it in lab, by the way, is to size separate DNA fragments. And ultimately, that's going to create a banding pattern on a gel. So let's talk about gel electrophoresis a little bit more. This is the schematic of a setup for a gel electrophoresis methodology. So essentially we have a box here. It's called a gel box. And inside of that box, we place a gel. So the gel is made up of usually like a 1% agar solution, which is a polysaccharide that derives from certain seaweed species. But basically it has the consistency of Jell-O, like kind of a thick Jell-O, because you can like hold it in your hand without it breaking. But it's a thick Jell-O. So anyway, the gel is casted using a gel cast into a sheet. So we have a sheet of Jell-O, sort of. Jell-O consistency material. And that gel has these little holes that are punched out through it. And these little wells here is where we would load samples of DNA. And we would set that gel into the box in such a way that the wells that house the DNA are going to be at this end, the negative electrode end. There's a negative electrode end and a positive electrode end on the other side, and it will be connected to a power supply. Now, because DNA is negatively charged overall, whenever we turn that supply box on and an electric field runs through the gel, the DNA is going to migrate this way, because it's going to migrate towards the positive electrode because it's negatively charged. Remember, positives and negatives attract. So samples are loaded into the negative electrode side. We're going to let that gel run for about 30 minutes, maybe 45 minutes, depending on how large our DNA fragments are. But what we're going to see is fragments of DNA on our gel. The larger fragments are going to stay towards the top, and the smaller fragments will be towards the bottom because the smaller fragments move much faster than the large ones. And I want to show this video to help elucidate.
When I was a young kid, I was really interested in genetics. Well, I didn't really understand genetics. I kind of thought that when two organisms had a baby, the baby was just this blend of the two. Yeah, that's a misconception. But I really saw genetics in action with my guppies. Guppies are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which means there are no eggs, like many other fish. And second, they have a lot of babies. They also eat their babies, but I don't think that's especially cool. So as you can see, that is not part of my cool fact list. Anyway, when my surviving baby guppies grew up, they would have all kinds of cool traits. These traits were carried by their DNA, their genetic material, which is found in their body cells. But sometimes I would forget which mother was the mother of the baby guppy, because there were several mother fish in the tank, and I wanted to keep track of inheritance in my guppy notebook. So what would have been very cool to have at that time? Some biotechnology. Biotechnology is the merge of biology and tech, and it's constantly changing. It includes topics such as PCR, cloning, and genetic engineering. It's also an awesome field. We're going to talk about one of the biotechnologies that could have, well, potentially, helped me determine the genetic relationships of my guppies. If, as a young kid I had access to it, although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate molecules based on how big they are, their size, and it's especially useful with DNA. Let's look at DNA real quick. So here is a guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell, and if you were to zoom into the DNA, here is a nucleotide, which is a building block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis, which again separates molecules based on size, relies on the fact that DNA molecules have a negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which, if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I use the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight. They take more time to make it across the gel when you compare it to shorter DNA pieces which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for relationships. Let's say that we have 3 mother guppy samples to view, and these are the only possible mothers from the fish tank. Which one appears to be the most related to the offspring in this case, and has a high likelihood of being the mother? Well, this one. But we can't be 100% sure with this. It would be helpful for me to know the father guppy sample too, because this will give you more insight. But if these are the only fish in the tank, it's a very high likelihood with this case. Also, you can use something called a DNA ladder. You can buy them from various science material distributors, but a DNA ladder is not what it sounds like. It's basically a sample that has known fragment sizes. So if you run it in the electrophoresis machine, you already know the fragment lengths. Let's say this DNA ladder only had 3 bands, which is not usually the case. Since it's a DNA ladder, the base pair lengths are known. They are 500 base pairs, 1,000 base pairs, and 1,500 base pairs. Think for a minute. Where would they fit in? It would look like this. You can use this now as a reference to give estimates of how large the other fragments are when they're run alongside it. And if you want to be closer to the value, you can use a semi-log graph, something to look up. So why do we care about gel electrophoresis? It's not likely I'll be actually using this with my guppies anyway, right? Well, perhaps. But gel electrophoresis is often a step used in determining relatedness with different species, which helps scientists better classify organisms. It's also used as part of DNA fingerprinting. DNA fingerprinting is a way that one can identify someone's DNA, which can be very helpful if you're trying to solve a mystery involving a crime scene. If you have a DNA sample from a crime scene, you can go through the steps of gel electrophoresis to compare it to the suspect DNA to see the likelihood of a match. In fact, you can take the results from gel electrophoresis and isolate genes of interest by something called southern blotting. Definitely something to look up if you're curious. Gel electrophoresis is one of many awesome tools in biotechnology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so anyway, that video hopefully demonstrated to you exactly how gel electrophoresis works. You saw that the DNA samples are loaded into the negative end, and then as the gel electrophoresis box is turned on, that electrical current forces genetic material to move towards the positive end, resulting in fragment patterns that are based on size, where the larger fragments are towards the negative electrode and the smaller fragments are towards the bottom electrode, or the positive electrode. So if we look at this over here, this will demonstrate, similarly to the video, an example of an application for this gel electrophoresis. And this here is a paternity test. So paternity tests are utilized to help identify the father of a child. So in this case, we have the mother's DNA, we have the child's DNA, and then in these two wells, these lanes, we have two potential fathers. So by analyzing the banding patterns of the gel electrophoresis, we can establish which father is the father. Okay, so in this case, the child's DNA, we will compare it to the mother's as well as the potential father. So this band of DNA was obtained, and as it shows in purple, from the mother. Now, in reality, our band would not be colored unless you were doing a fluorescence. There's a special kind of gel electrophoresis which utilizes fluorescent primers that could then potentially show you where it came from, mother or father, but in some they don't. But again, we're just trying to talk about in general. There are some advanced concepts in gel electrophoresis that we won't really cover in here, so we're just going to discuss the basics. But this band, as it appears on the gel, it was established from the mother. That's because these two bands are the same size. Remember, all of this DNA from each sample, mother, child, and male 1 and 2, were cut with the same restriction enzymes, and thus, because their DNA is different, they are going to have slightly different banding patterns. So these two bands here did not come from the mother. So that means they had to have come from the father. And as you can see, male number 1 is the only one that has those two bands. So that establishes then that male number 1 would have to be the father of this child. There are some other additional potential uses. So besides paternity analysis, we could also utilize the same procedure with analyzing DNA found at a crime scene. We can compare different organisms, even those from ancient times, if we have fossils that have retained DNA. We can use it for medical diagnoses for some genetic diseases as well. This is another example. This is showing you crime scene DNA. Here we have crime scene evidence. So we can collect the crime scene DNA from that evidence, whether it be blood or scrapings from underneath the fingernails sometimes, because they may have suspect DNA under them, because one of the most common things you'll do to protect yourself is to start scratching or throwing your hands out, doing something. So that's a common area where suspect DNA is looked for. Then we have suspect A and suspect B. So it shows it shows repeats here, and this is the more modern way of doing things, and I have explained that in this slide here. So we'll go back to that picture in just a second. I feel like I put my slides in out of order here, but present day, we talked about past already, how we just use restriction enzymes on DNA. Now we do it a little bit differently, where we utilize these short repeating sections of non-coding DNA in order to create DNA fragments instead. So these short tandem, we call them short tandem repeats, or STRs, are things like GATA, GATA, GATA. And you might have 3 of those at a certain loci, whereas I might have 10. And so if we analyze enough of these STRs and compare them between one individual and another in or crime scene data or whatever the case may be, we can help establish, how similar DNA is from sample 1 to sample 2. So in this case, it shows you crime scene DNA. Our crime scene evidence shows 2 alleles. Remember, for every gene, you have 2 alleles. We have 12 repeats and 16 repeats. All right, so that is then demonstrated here. The 16 repeat would be here and the 12 repeat would be there, which makes sense, right? Because the one that's smaller, the 12 repeat, moved faster and thus got further along the gel. Next, we have suspect A. We have 12 repeats on both alleles, which then says we only have one fragment. And then we have 12 and 16 for suspect B. So comparing crime scene with suspect DNA, we would say that suspect B is a match. Now, another lane you see here are markers. We won't really cover this one. Basically, these are just known bands to help us compare from, the data that we created from the suspects in the crime scene. So basically, because these are known, we know that band is 14. We know this band is 10, so we can estimate about how many repeats this person has or this sample has in each band because we're comparing it to something that is known. Okay, so this video here, I'm hoping again that it will be loud enough for you to hear it, but it is a review of short tandem repeats. Again, short tandem repeats or STRs are just repeated small fragments of DNA that are, between like 2 and 9 base pairs or so.
A short tandem repeat, or STR, is a region of DNA composed of a short sequence of nucleotides repeated many times. Since the number of repeated sequences in a given STR varies from person to person, pinpointing these variants can be useful in DNA fingerprinting. As the name implies, the repeated stretches in STRs are short, only 2 to 10 base pairs long. For this reason, and because they are dispersed more evenly throughout the genome than the longer variable number tandem repeats or VNTRs, STRs are favored by forensic labs. Large databases of information on STRs in the general population tell analysts how much variation exists at any given STR location. That information can help forensic analysts determine the conclusiveness of a match between two samples. If 10% of people have the same number of repeats at a given STR, for example, finding a match at that site is merely suggestive that two samples are a match. If two sites with the same prevalence match, the odds drop to 1 in 100 that the similarity is coincidental. If 13 sites match, the odds that any two people would possess such a fingerprint are so small, about 1 in 10 trillion, that the result can be considered a definitive match.
Okay. So hopefully that helps show y'all how these short tandem repeats can be utilized. We know how many are at different loci, or what the likelihood of having 9 or 10 or 12 repeats are. So by using multiple loci and comparing them to different individuals, we can identify matches or how closely related organisms are. Okay, next, gene editing is a really cool tool that is relatively new in biotech, but it does just what it sounds like. It edits DNA. CRISPR is a very is one of the leaders in this field. CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. That's a mouthful, but it was discovered in prokaryotes as a viral defense mechanism. Cas9 is an endonuclease enzyme that identifies specific portions of DNA to be cut. And a PAM sequence must be adjacent to it in order for the process to be successful. But what you can think about CRISPR is a little tiny molecular pair of scissors, just like our restriction enzymes, that cut out specific stretches of DNA. Here is a illustration of CRISPR and Cas9. So here we have DNA, genomic DNA. We have CRISPR, which is made up of a guide RNA shown here. The Cas9 protein is in green. The PAM sequence is in orange. And then we have a matching genomic sequence for the portion of the DNA that we want to break. So essentially we are able to create a double-stranded break in DNA and remove a gene and ligate the DNA back together to completely eliminate a gene that we do not want. And again, we have I don't want to say mastered the art because there's a lot of work that still needs to be done with it, but we have tailored the art of this RNA protein complex, CRISPR-Cas9, to our needs and to help us to edit genes for human use. And this is one of those things that's still in clinical trials that are multiple genetic diseases are in clinical trials. I think CRISPR has 5 right now that are in clinical trials for curing certain genetic diseases. Next, stem cells and cloning. That's 12.2 in our text. So every cell in your body, as you know already, has a single copy of all of your genes, or technically 2 alleles, so 2 copies, but it receives a copy of all of your genes. And every cell in your body has the potential, therefore, to become a complete organism because we have the same DNA in every single one of our cells except sperm and egg. And cloning utilizes this potential. And there's 2 types of cloning that we can discuss in biotech, and it's reproductive cloning and therapeutic cloning. Reproductive cloning is where we desire to create a new individual. We are going to take a sheep and we are going to clone it to create a brand new sheep. Plant cloning is very common. Animal cloning is possible. It was thought to be impossible previously, but the first cloned animal, her name was Dolly, and she was a Dorset sheep. And how they cloned her is, she was the first clone. So her mother, they took an adult nucleus and placed it into an enucleated cell. So what that means is they took a cell that came from either her mother or father, and removed the nucleus, that means enucleated it, and then placed a nucleus within it from a different cell, from a different parent. And then they allowed that donor cell to starve for a little while. And that just means you're not giving it any nutrients. And that forced that cell to go into G0. Remember G0 phase when we talked about mitosis. And then they were able to signal for it to initiate development after it went into G0 phase. And the way they did that was to introduce it into a womb, a uterus. All right, so these are the 2 forms of cloning. We just went over reproductive cloning. That's where they take G0 cells from an animal that they want to clone. They remove a nucleus from it. You can see the nucleus is removed. And they place that nucleus into an egg cell that has no nucleus. So there's no nucleus in it. They took it out. So they took the nucleus from one cell and put it into an empty cell that happened to be an egg cell. And then they allowed it to sit in a culture dish until it became an embryo, which is just a collection of cells. It's probably like a blastula phase, I would presume. So it's probably a ball of 100 cells or so. And then they put it into the womb of an adult female sheep. So this is basically in vitro fertilization in animals. And then that implanted embryo was birthed by the surrogate mother at a later date. And so that was the clone being born. So that is reproductive cloning. The goal here was to create a whole new sheep, whereas in therapeutic cloning, we are only trying to design cell types. So instead of having a surrogate mother and all of that good stuff, we're taking G0 cells and we are placing the nucleus of those G0 somatic cells into an empty egg cell and then just allowing them to be cultured into a petri dish. And so those cells can become, muscle cells, they can become blood cells, they can be nervous system cells, and so on and so forth. They become specialized. over time. But our goal here isn't to create another human or create another sheep. We're just trying to make cells of a specific type. Okay, so let's talk about animal cloning. So farm animals with desirable traits are commonly attempted to be cloned. Sometimes endangered animals are cloned because we don't want them to become extinct. Now, we will not find any cloning of humans, however, because there are ethical constraints there and legal limitations. No federal funds can be used for any experiments to clone humans. And this is for a few reasons. So for the obvious reason, cloning a human just is, I guess to me anyway, in my opinion, is pretty unethical. But the bioethics there is just, I don't think that's okay. Think about the pitfalls of it. So if we are able to clone humans, and I have a clone of myself, hypothetically, what if she commits a crime and then they think it's me? Stuff like that. I mean, there's a lot of other different examples, and we will talk about bioethics in our discussion board in lab, so I'll leave that for some food for thought. But right now in the United States, there are no federal funds that can be utilized for experiments to clone animals, and there are limitations legally that inhibits us from doing those experiments as well. So some of the reasons for that, besides bioethics, is that not all cloned animals have exhibited abnormal aging, but some do. So, for example, Dolly, the sheep that we just talked about being cloned, she had to be put down due to having severe arthritis. And there was another issue she had that I can't recall from the top of my head, but I know it's in your textbook. And she had only lived about half the lifespan of a normal sheep of her species. So additional research needs to be done to address the challenges of animal cloning. But the products from cloned animals are just as nutritious as the products from non-cloned animals. So that's something to think of as well. There are some positives to animal cloning, and there's also some downsides. People can abuse the system. All right, therapeutic cloning. The therapeutic cloning type of cloning is where we are ending up with desired mature cells, not whole individuals. So we're trying to get blood cells, or we're trying to get nervous system cells, or we're trying to get muscle cells, etc. So we are able to use these cells to treat human illnesses sometimes. So, for example, if someone has a spinal cord injury that has paralyzed them, if we can get these cells to become these somatic cells to become nervous system cells, and then insert them into the spinal cord where the damage is, maybe we can fill in the gap where that damage is and give that person their ability to move back. These are potential things we could do. Anyway, this process can be carried out in a few different ways because there's different types of stem cells. So we talked about cloning already when it comes to inducing cells to become certain tissue types, but there's also a couple others. The first one that we'll discuss is using embryonic stem cells. As you can probably presume, they stem from embryos or sometimes umbilical cord blood. The cells found here are considered totipotent, and that's really cool and really important because they can become any cell type. Think total is the way I think of it. But this has a lot of bioethical concerns because you're utilizing cells that are of an embryo. So, is it ethical to take embryonic stem cells and utilize it for human benefit like that? Some people will argue yes, and some people will argue no. So, for example, if someone gets an abortion, they could donate that embryo to science, and that embryo cells could be utilized to help create treatments for certain diseases. So, something to think about. But is it ethical? Is it not? It's something that people argue. And again, you do have a discussion board that talks about some of these topics where you can voice your thoughts and your opinions. Adult stem cells are another type of stem cells. They are considered multipotent. They cannot become just any type of cell. It's limited. So essentially, they can only become they can only become a limited number of different types of cells. So you have a lot of like constraints there with being able to use those versus embryonic stem cells. However, there's not the bioethical concerns with using adult stem cells. Now, there is a possibility to overcome this limitation. Some scientists have been able to coax adult stem cells into behaving more like embryonic stem cells by adding certain genes to them. So that's really interesting. Another thing to look up if you're interested, which is basically what this is, and I'm not sure if I'm spelling this right. And I think this is relatively a recent advancement in stem cell research, induced pluripotent stem cells, but it's again where scientists can coax adult stem cells to behave like embryonic stem cells. And I first remember hearing about it when I was in grad school, so I don't know if that's when they started creating them, but either way, relatively recent advancement in that technology field. All right, so utilizing biotech, what kind of products have we gotten? So we have made transgenic bacteria. So transgenic bacteria have genes in them that come from different species. They're grown in these giant vats called bioreactors, as you can see here. But basically, they are going to contain genes and therefore produce product that we want them to. Insulin was one we already talked about. They can also grow growth hormones, tissue plasminogen. They help create the Hep B vaccine, clotting factor 8. All of these different things help to either prevent or treat human diseases. GMO bacteria have been designed to clean up oil spills. So, for example, here we see bacteria being grown in cultures, and these particular ones are, I believe this one is from an oil spill cleanup variety bacteria. But there's even some GMO bacteria that have been designed to create frost-resistant strains, not trains, of plants. But, there's certain bacteria that are found specifically on certain plant species. We can harvest those bacteria, grow them in a lab, edit their genome, release them back onto the plant, and now that's helped create frost-resistant strains. Transgenic plants, corn, cotton, potatoes, soybeans, all of those are very commonly genetically modified. They can be genetically modified to be insect or herbicide resistant. So insects won't be able to destroy crops, and herbicides will not affect them. So that means then that we can spray our cotton field or our corn field or potatoes or soybeans, and that herbicide we spray will not kill the crop. It will just kill the weeds. And that weed killing means that we don't have to till the land as much to physically get rid of them. It means there's less soil erosion and just overall better growth, because weeds basically suck the nutrients from the soil that the cotton, corn, potato, or soybeans could be utilizing. Some other transgenic plants, we are able to utilize biotech to help improve shelf life. Arctic apples, utilizing gene knockout, which means we deleted a gene. We were able to create a variety of apples that don't brown. Innate potatoes, another similar kind of biotech, RNA interference. It downregulates genes, and it does so for the genes that cause bruising in potatoes. So you can see here a picture of a conventional versus an innate potato, whereas this one here doesn't bruise such as the conventional one does. Here's the Arctic apple at the top. So you can see all the bruising and the browning in the apple up here versus down there. Plants, they have been improved by increasing their nutritional value. Think rice and soybeans. Rice, basically a really starchy, just not super nutritious item on the menu, but they have been induced to be able to have protein within the rice, which is an important source of nutritional value. Soybeans, they have been modified to have like I believe it's monosaturated fats, rather than more complex fats within them that are easier to digest and thus leads to better health in the community, less heart disease, for example. They have been designed to have increase in productivity. So we are able to design leaves that are less inept to water loss. So we don't have to water them as much. We don't have to water plants as much. We save water. We save energy because we have to use energy to run watering systems like fossil fuels and gas and electricity. It also means that if we don't have to water plants as much, then the soil is less susceptible to fungal growth, which then means we have healthier plants. I have a garden outside, and I overwatered them because this is my first time gardening, and there was literally piles of fungus growing on the ground. It was disgusting. I had to use a hoe and basically hoe it out. It's kind of like look up a picture anyway. It's like a flat shovel, sort of, that I had to scrape all the ground and get all of that out because otherwise all my zucchini and my squash were growing and smushy and had this black and white fuzzy stuff on it, which I knew was mold because I overwatered them. It was gross. But anyway, so another benefit of GMOs. We can also increase productivity in plants by designing them to take up more carbon dioxide, which is a necessity for plants. Now, that also enables plants to be grown in various climates. So we can grow crops that in arid communities that don't have, or very dry, like in the desert, it's very, very dry there. If we have plants that have been genetically modified to where their leaves are taking up more carbon dioxide and preventing water loss, we can grow plants in more drier climates than we previously could. So that means we have less needs to import, less needs to ship. It aids the economy that way. We can grow things in America and not have to import them from wherever they were previously grown in. All right, so some plants are used to produce human hormones, clotting factors, vaccines, and antibodies as well. Moving forward to animals, transgenic animals have been created that help us to create larger fish, cows, and pigs by inserting growth hormone genes. Previously, transgenic pigs were used to supply transplant organs for humans. That process is called xenotransplantation, where we take an animal organ and put it into a human. Think heart valves. Those were previously made by pigs. Mosquitoes. Mosquitoes are a vector for a lot of human diseases, and they have been genetically modified with this quote-unquote kill switch that produces proteins that kill some of their clutch of offspring. And so, therefore, we reduce the population size that way of mosquitoes that can contain West Nile virus, Zika, malaria, Eastern equine encephalitis, yellow fever, dengue fever, some of these deadly and some of them no cure. So very important benefit of biotechnology. Another thing that animals have been used for in biotech is farming. Notice that it has a pH at the beginning. This is where we use transgenic farm animals to produce pharmaceuticals in their milk. Vaccines, medicines, in some circumstances will be utilized or will be produced utilizing that process of pH farming. Transgenic animal mice are also routinely used in medical research. The way that they are commonly used in medical research is that they can have genes that are known to cause human disease will be cloned into mice, so then we can study the effects of the disease or even test treatment on the mice. Lastly, 12.4 is genomics and proteomics. So genomics is the study of genomes, the entire genes. That includes humans or other organisms. We can analyze coding sections and non-coding sections. The Human Genome Project was a 13-year effort. Multiple scientists were kind of in a race to try to finish it. But essentially it was to establish the entire genome sequence of humans. They found that there are many small regions of DNA that vary among individuals, and some individuals even have extra copies of genes. And these differences may have no effect or may increase or decrease susceptibility to diseases. So here we have A, B, C, and D. If we look at A, this is a normal chromosomal DNA. We have gene A, B, and C. If we analyze gene A, the gene, excuse me, is TACGGG. If we were to then predict the mRNA that would stem from that, it would be AUGCCC. Then we could use our genetic coding table to figure out what two amino acids these would translate into, and we see methionine and proline. Now, if we have a variation in the order of the bases in a gene due to a mutation, such as here, we now have, instead of TACGGG, we have TACTGG. Well, that will alter the mRNA transcript, that mutation here. So instead of AUGCCC, we now have AUGACC. And then if we analyze the amino acid that would be added via the genetic code, we would have methionine and threonine. That would then change the three-dimensional shape of the protein and thus could alter its functionality. It could increase the functionality, decrease the functionality, and sometimes not have much of effect at all. It just depends on how severe the change is. All right, C. This is a variation in the bases within an intergenic sequence due to a mutation. So here we have gene A, B, C. These would be introns. Now, obviously, this would not really have most likely an effect on the organism because it is an intron. And introns, remember, are in the way. They get spliced out. Now, that's not to say that it couldn't have an effect on the organism as a whole. It's physiology. But we won't go into that because it's a more advanced topic that would be covered in a higher level bio class. So we'll leave it at that. Lastly, variation in gene copy number. So this individual has 2 copies of the B gene, and that could or would increase the functionality of that particular protein product because there would be more of it being made because they have 2 genes rather than 1 for it. Okay, next, genome comparisons. These are clues to evolutionary origin. So remember, we can analyze how closely related genetically or evolutionarily organisms are by analyzing how similar their DNA makeup is. So again, like chimpanzees are around 98, 99% alike. Humans and mice are around 85% alike. But even, bacteria, we still share some genes with bacteria because ultimately all organisms, via the concepts of evolution, stem from one organism long, long ago. We'll get to that in another chapter. All right, so if we compare human and chimp, chromosome numbers 22. Among the genes that differed were several that may have played a role in human evolution, such as speech, hearing, and smell. Comparing genomes may be a way of finding genes associated with human diseases as well. Again, think about if I have a disease and you don't, if we compare our DNA, we may be able to figure out which one of those genes are associated with said disease, especially if we have a really large sample size of people that have the same disease, and then compare it to a large sample size of people that don't. Next, proteomics and bioinformatics. Proteomics is the study of your proteins. It explores structure and function of proteins and how they interact to produce traits. It's really important in drug development because everybody's genome is different. That means everyone's proteome is going to be different, and proteins interact with each other. And therefore, if we introduce a drug into an individual, it can have a different reaction than it does in me versus you. Next, bioinformatics. This is an application of computer technology to study genomes and proteomes. So that really helps us analyze a large amount of data and see if we can find significant patterns. Lots of money in that field. All right, that was chapter 12, y'all. Hope you enjoyed it.